All right, I'd like to thank you all for joining us. My name is Steve Feeney. I'm a technical application scientist here at CDI, and we'll be beginning in just a few seconds. All right, today we'll be covering the iCell cardiomyocytes, functional human cardiomyocytes derived from iPS cells. We'll be going over a little bit of a general overview of their characterization as well as some of their utility. But before we get started, I want to explain, for those of you new to the webinar series, just how you can ask some questions. As we'll be going along, I'll be going fairly quickly because we have a lot of data and a lot of great customers and collaborators that are doing a lot of wonderful things with our cells. So you can go ahead and you can ask questions as we go along, and we'll get to them at the end. As you'll notice on the top of your screen, you should see a Viewing Cellular Dynamics desktop in green right at the top. So if you place your cursor over top of that interface bar, it should drop down with some options, one of which is Q&A. If you don't see Q&A on the bar, there's a drop-down arrow on the far side, and if you click that, it will be one of the options there. This should prompt you with a little instant messenger interface where you can write in your question and then click send, and as we go along, we'll collect those and go over them at the end. So what we'll be talking about today, we'll go through a quick overview of CDI and who we are and what we do, as well as the iCell cardiomyocyte product, just an overview of the product itself, what you'll get. We'll go into the basic characterization of the product that we've done, and then we'll go into some iCell cardiomyocyte utility, from biochemical toxicity screening, functional interrogation of relevant biology and reporter assays, disease modeling, more of your discovery, and then we'll wrap it up with a quick summary. So who are we? Cellular Dynamics International is the world's largest producer of human iPS cells and iPS-derived cell types. Currently, CDI has about 107 or more people now, and we really have three core competencies. And those are the creation and culture of human iPS cells, genetic engineering of the iPS cell lines, and manufacture of human iPS cell-derived cell types. Those are really our three main strengths that bring you the iCell products. And of those products, we currently have available the iCell cardiomyocytes, which was our first product and we'll be talking about today, the iCell endothelial cells, which were launched in Q3 of last year, and the iCell neurons, which were launched in Q4 of 2011. We also have the hepatocytes, which are on a pre-commercial evaluation period, and the MyCell custom services, where we can do custom iPS reprogramming from just a simple blood draw of one of your patients or tests. So what are the key characteristics for adoption of iPS technology? And really, we view there are three main key characteristics that we have to hit. And those are quality, purity, and quantity. Now for quality, we ensure that our cell types exhibit key cellular characteristics, and they're able to recapitulate normal human biology. And they do so in a reproducible manner. For purity, this becomes a very big issue. We can select our cells to get very, very high purities in the upper 90s. And this becomes important so you know what you're studying. With a lot of primary cells, for example, you get your target cell, but you also get other cells. You get your fibroblasts and contaminating cell populations. And oftentimes, these contaminating cell populations proliferate. So your window of testing and culture can be shortened dramatically because your non-target cell population is proliferating and overtaking your target cell population. And finally is quantity. We make sure that we can supply sufficient quantities in order to support high-throughput drug screening and safety testing. And this means that for the cardiomyocytes, we have the capabilities of producing 1 billion cardiomyocytes per day. So in our iCell product portfolio, right now we currently have 5 products. The iCell cardiomyocytes, which are currently available, the endothelial cells, which are also available, and the iCell neurons. The iCell hepatocytes are currently in their early access phase, and we have down in the bottom right corner the custom iPS cell services. So just a quick overview of what you get with the iCell cardiomyocytes and what they are. So they are pure human iPS-derived cells. They're cryopreserved and they're ready to use, and they're available in virtually unlimited quantities. So you no longer have to wait while you're differentiating stem cells or differentiating progenitors. You can simply take your cells, thaw them, and use them. And we also offer a full product solution. So this means that with each order, you get not just the cells, but you also get the plating and maintenance media to maintain them. And you get some protocols to thaw the cells and get started. And then they demonstrate the normal human cardiac biology, electrophysiology, and toxicity responses, which we'll cover in just a few slides, and the broad platform utility for discovery and preclinical development. Now, as you've probably noticed by now, down in the bottom right-hand corner is a video of the iCell cardiomyocytes. And when they're in culture, they do contract, and that's what you're seeing here, is the iCell cardiomyocytes contracting in culture. Now, in the bottom left hand is a purity. So CTNT, which is cardiac troponin T, is just demonstrating that for this population that it's 99.3% pure, with an image on the in panel B just showing the sarcomeric alignment and the fact that in culture everything is a cardiomyocyte. This next slide just kind of demonstrates that for a product that's only really been on the market for the last year, year and a half, that they're already being used in a wide number of ways and currently going out for publications. So you can just see that a number of our different collaborators are using our cells in a number of different ways, and they're already providing publications. All right, now we'll begin in with the iCell cardiomyocyte characterization. And we'll start with the genetic characterization. So on the left here, I have a transcriptome analysis, which was done by Roche. And using their algorithms, they categorized the genes into 3 different categories: stem cell genes, transition genes, and cardiomyocytes. And really the key things to see here is when we start from our stem cell population, as you'd expect, we have very high stem cell gene expression and very low, essentially none, transition and cardiomyocyte gene expression. And as time goes on, the stem cell genes shut off and they remain off. The transition genes turn on during differentiation and then shut off again and remain off. And the cardiomyocytes turn on during differentiation, and then they peak and they remain. And so really what I want to show here is the first 30 days here in yellow is our differentiation. And as you can see, during the differentiation, the cardiomyocyte genes peak. And here at day 30 is where we freeze down the cells. Now, for the sake of this experiment, we maintained them in culture for an additional 3 months. And what you were able to see is that the cardiomyocyte genes remained essentially completely stable. So we have a very long-term stable gene expression. So now the next thing that Roche did was they took our cells with ambient RNA and they rank ordered them from highest to lowest and just saw how our cells compared at day 28 and day 120. And the 2 things to take away from this top graph here is first, between day 28 and day 120, that the blue and the red lines overlap. And as you can see, there's a very strong correlation between the ambient RNA and the iCell cardiomyocytes. So next, they took primary cardiomyocytes that are currently available on market. We've got the CellProgen and the PromoCell. And they did the same analysis, and they rank ordered them, and they overlaid them. And as you'll see, the primary cardiomyocytes compared to the ambient RNA didn't overlap as strongly as the iCell cardiomyocytes. Now this prompted a kind of tongue-in-cheek quote from the investigator at the time that our cells were more human than human, which, as a provider of the cells, is a very good marketing tool, which is why you'll see it on this slide here. Next, from the gene expression, we go into the protein expression. On the top panel in panel A is just cardiac troponin T and alpha-actinin, just once again showing the very high purity in the population, up towards 97.4%. In panel B is the cardiac troponin T, showing that it's prevalent in all the cells. In panel C, we have alpha-actinin, and the interesting thing in panel C is, as you can see, we've got the striation showing the organization of the myofibrils. Panel D is cardiac troponin T and MLC2V, and this just demonstrates that it is a mixed population. So we have nodal, atrial, and ventricular cells in here. Panel E is, again, cardiac troponin T in green, with connexin 43 for gap junctions in red, showing that they form along the edges as you'd expect to really enable that cell-to-cell communication. Additionally, with the N-cadherin for the, you've got it right along the edges as you'd expect. Going into the iCell electrophysiology characterization, you can see from the left-hand side we have all of the expected ion currents from the depolarizing sodium and calcium currents to the repolarizing potassium currents, ITO and IKR, as well as I-funny for pacemaking, and IK1 to set your resting membrane potential. Going into the spontaneous action potentials, which is really a unique feature of cardiomyocytes in general, as opposed to just a transformed line with a specific channel, is you've got all the channels, but you also have the cardiac action potential. And the iCell cardiomyocytes have all three cardiac phenotypes from atrial, nodal, and ventricular-like. And we've got those characterized to show just their general expected fallout in the population. And then here on the bottom we have the GPCR signaling pathways, beta-adrenergic isoproterenol, alpha-adrenergic phenylephrine, and the muscarine receptor, carbachol. This down here on the bottom was done via MEA, so a field potential recording just to get a population response. Next is the iCell cardiomyocytes. We have metabolism and contraction just to show that our cells are very metabolically active, as you'd expect, and that we have calcium-dependent contractions, as shown via the ion optics. Both the seahorse metabolism data and ion optics we'll get into a little more depth later. So next we'll go into the iCell cardiomyocyte utility. We'll cover a number of different platforms that our customers and collaborators have been using, and just give you an overall idea of some of the things that can be done with the iCell cardiomyocytes. First is the electrophysiology. Our cells have been used on manual patch clamp, automated patch clamp, and MEA systems. And as you can see, you can not only investigate the individual ion channel that you're looking for, but you can look at and see the expected drug response in the action potential as well, and kind of get a feel for all of them in once. Here on the left-hand side, we have IKR, and as you add E4031, you get the expected IKR block of a field potential prolongation, with the ability to induce EADs as well. In the automated patch, here we're looking at the calcium channels, and as you block with nifedipine, you'll get the expected action potential shortening. And finally, we go into the MEA, where you can really look at more of an organotypic investigation, because you have a population of cells now as opposed to an individual one. And you get the normal field potential, as you'd expect, very similar to an EKG, as you have your depolarization and your repolarization. And again, with nifedipine, you get the shortening of that field potential duration, and in a nice dose-dependent manner. Next, going along the same lines of arrhythmia, we'll go into the ASEA's Roche RAS Excelligence system. This is an impedance-based system that comes in a 96-well plate, and on the bottom of the plate, it's covered in gold electrodes to measure impedance. So with the iCell cardiomyocytes, as they contract, they physically change shape, and you can get you can measure out as a difference in impedance as they contract. So what was done by Roche in this one is you've got your stable DMSO and aspirin negative controls. You see a very nice, stable, repeatable phenotype as it's beating consistently, and then they applied a number of cardioactive compounds. And as you can see, the compounds had very different effects, but very arrhythmogenic. So what they did next is they made a predictive pro-arrhythmia score, and they rank ordered the compounds based on the highest predicted pro-arrhythmic score to the lowest. And they compared this to their data in vitro from herd tests, QT tests, and then animal arrhythmias, or in vivo arrhythmias. And the key things to point out here is the compounds that came up as negative in vitro were positive in the iCell cardiomyocytes and positive in the in vivo tests as well. Compounds that were positive in both were again detected by the iCell cardiomyocytes. and compounds that were positive in vitro but negative in vivo were also detected by the iCell cardiomyocytes as having a very low risk. And again, the negative compounds that were negative throughout were negative in the iCell cardiomyocytes. So this really shows the predictive value of the iCell cardiomyocytes and their utility. Next goes into just cellular toxicity. And this really demonstrates that any of the over-the-counter kits that you can get from Promega to do a very straightforward viability or different mechanistic investigation, you can do with the iCell cardiomyocytes. They're very amenable to many different platforms and many different assays. And here with Cell Titer Glow, we've got a number of cardiotoxic compounds, and you can see the expected toxicity throughout. But you can also take that general viability and go to specific endpoints, whether it's oxidative stress, apoptotic activity, HDAC activity, or proteasome activity. This means you can really get a mechanistic insight as to why your compounds are toxic. It also provides a cell type specific toxicity. Here we have iCell cardiomyocytes compared to a cancer line, K562. And when you hit them with SAHA, a cancer drug, you can see that the apoptotic activity in the cancer line increases and the viability strongly decreases, but you don't get the same kind of toxicity in the iCell cardiomyocytes, as it's a completely different cell type and you get a cell-specific response. Going into contraction, this work was done by Pfizer on the ion optics system, which can measure the calcium transients as well as contraction as determined by edge detection. And here, as we block the calcium channel with nifedipine, you see a dose-dependent decrease in the calcium transients. But you also see a decrease in the contraction, as calcium is critical to the cardiomyocyte contraction. So with a number of different compounds, this really gives you the ability to investigate positive and negative inotropy. As you can see that isoproterenol and BK8644 increase the calcium and increase the contraction strength, whereas the calcium blockers, nifedipine, verapamil, and diltiazem decrease the calcium transient and decrease the contraction strength. Next, staying on the same theme as calcium handling, brings us to the Flipper Tetra system, which can measure the fluorescence intensity of calcium transients in autonomically beating cells. So this work was done for us by Molecular Devices. It's a high-throughput 384 plus platform, and you can really use the calcium transients as a surrogate for beating. So here we have our control and epinephrine showing the increase of beating, as well as a number of other chronotropic drugs showing the increases and decreases respectively. Now next you can take this and go a little bit further, and the calcium transients can really reflect electrical activity at the membrane. So here we have cisapride, which is a hERG blocker, and as you can see from the calcium transients, an increase. And with their Peaks Pro software package, you can do a number of complex analysis on the calcium transients. Next, we'll go into energetics, starting with the Seahorse Biosciences instrument. This instrument can measure oxygen consumption rate and the extracellular acidification rate. And from these, you can really start to interrogate the energetic and mitochondrial functions. So on this graph, the first section we have here is just baseline conditions. So this is our baseline respiration. We followed it up with an injection of oligomyosin, which blocks ATP synthase, so we can really get what our ATP production is from the mitochondria. Then we hit it with FCCP, which uncouples oxygen consumption and ATP synthesis to really see what our maximum respiratory capacity is. So if the cells were working at 100% efficiency, this is what they could turn out. And that gives us an interesting parameter to investigate as our spare respiratory capacity, just the amount of capacity that the mitochondria have under duress before they're completely maxed out. Next, we hit it with rotenone, which blocks complex 1 of the mitochondrial electron transport chain. And so we're left with the non-mitochondrial respiration. And as you'll notice, we have a small gap here, and that can really assess the efficiency of the mitochondria and the protein leak across that membrane. So continuing with bioenergetics, we have from Molecular Devices on the Image Express microreader, just a JC10 readout of the mitochondrial membrane potential, showing both healthy and cardiomyocytes treated with antimyosin A. So as you can see, we have a large number of mitochondria showing up here in orange that are not present after the treatment with antimyosin A, which blocks complex 3 in the mitochondria. So we can then go in and analyze using the Image Express software and quantitate just to get our dose response of antimyosin A and the bioenergetics of the cells. Now, the Image Express can be used for a number of other applications as well, and here we used it to read out our transient transfection. So we prepared a plate of iCell cardiomyocytes for 4 days, replated them in 96 well plates at 40 and 80,000 cells per well, and transfected with a cre-luciferase reporter for 3 days post-replating. These cells were then cultured for 18 hours and induced using isoproterenol to stimulate the cyclic AMP pathway and measured out. And what you can see from here is a very nice large fold induction, but also that the iCell cardiomyocytes can be transfected with different reporter assays, with different plasmids to really start to investigate your discovery targets. This opens up a wide variety of applications that you can use these cells for. Now, another discovery application is disease modeling. So for this example, we'll use cardiac hypertrophy, which is simply an increase in cell size. So as you can see from the image here, the small heart becomes the large heart, but there's a lot of involved pathways in the process. And you get an increase in protein synthesis, in cell size, and fetal gene expression. For this assay in particular, we stimulated using endothelin-1, and we measured NPPA or AMP and NPPB or BMP as an endpoint. So here on qPCR, you can see we had a nice fold induction with endothelin. Again, with microscopy, we had a nice fold induction. And with flow cytometry, measuring BMP, we had another nice large fold induction. But the key point is, across all these different platforms, you're getting the same EC50. You're getting the same results. So it's really platform agnostic, but you get to investigate a lot of different targets. So just in summary, the iCell cardiomyocytes, they can recapitulate in vivo human cardiac myocyte biology. They provide an ideal tool for interrogating relevant cellular physiology, and they're really platform independent. You saw through these slides the wide variety of platforms that our customers are using the cells on and the different results that they're getting, and they're available on demand. So whenever you need the cells, they're in a cryo vial, you can pull them out, you can thaw, and you can begin. So where can the iCell cardiomyocytes be used? So on the bottom half here, I have essentially the drug discovery pipeline, starting with your target ID validation, your hit identification, going into your lead optimization, your toxicity testings, and your clinical testing. And with the quantity and purity of the cells, you can really go in to investigate your different mechanisms. You can go into do your toxicity study, and you can really have a more relevant human model earlier in the process. But with that more relevant human model, you can move it even earlier into drug discovery and start using it as your target to start identifying your hits and start identifying potential compounds, and really essentially fail sooner to move the selection process earlier in the process. And with the overall potential of IPS technology, you can also go from the clinical and find a disease phenotype that you're looking for and bring it back into your discovery process through reprogramming services, through the MyCell reprogramming services. And with that, I'd like to thank you for joining our webinar. We'll go over a few of the questions, but if you have anything that's not covered here, please feel free to visit our website, www.cellulardynamics.com, or feel free to email support@cellulardynamics.com, or call us at 608-310-5100, or toll-free 877-310-6688. And with that, I'd like to get into a few of the questions that we received during this procedure. All right, so first, what is the optimal time post-plating for running these assays? The optimal time can really depend on a number of factors, kind of your seeding density, how well you thaw. There's a few little handling issues. But for most of our assays, we recommend between 4 and 7 days, with some that have a pre-plate step in there taking a little bit longer. One is, what is the difference between plating media and maintenance media? The iCell cardiomyocytes do come with 2 separate medias. The plating media is optimized more around just a general cell attachment, whereas your maintenance media is for more longer-term cultures. So you don't want to keep your cells in the plating media for long periods of time. Next is, do you have any data on the effects of antibiotics on the growth of iCells? I should point out that our maintenance and plating medias do not have any antibiotics in them, and that's more for the customer to not have to worry about any influences of them. We do not have any hard data at this time of specific effects. However, we just as a rule of thumb in-house, we just don't use antibiotics. When we have had to use them just for demo purposes, we've used gentamicin. What is the transfection agent used for the transient transfection with GFP plasmid DNA? So in the slide previously with the Cree luciferase, I believe it was the Mirus transfection reagents, Transit LT was used. And with that, we're going to wrap up our webinar, and I'd like to thank you for joining us. Again, please feel free to contact us and reach out at support@cellulardynamics.com. And thank you.